Okay, I have one last short video to wrap up genomes. So one of the things that I talked about in the earlier videos is the idea that we can use the human genome data, we can compare that with other organisms. And that's actually very, very useful. So we've actually sequenced the genomes of many other organisms. In fact, the first genome that we sequenced was actually from a bacteria called Haemophilus influenzae. That was the first one we sequenced, and bacterial genomes are very, very easy to sequence. Most bacterial genomes are several thousand genes instead of tens of thousands of genes, and the DNA is much simpler because bacteria have a single chromosome, a single loop of DNA. And so we've actually completed a lot of different organisms. So for example, here, these are just some of the things that we've sequenced. We've done prokaryotes like bacteria. We've done eukaryotes, so for example, yeast. We've done fruit flies, mosquitoes, nematodes, those roundworms, mice, pufferfish. We've done plants like thale-cress, which is a plant called Arabidopsis, which is used a lot in genetic studies, and crop plants like rice. What this graph is actually showing you is something we kind of expected, and we were able to demonstrate this, that if you look at the size of the genome, so the size of the nucleotides in that organism's genome, in general, there's this overall trend where as the size of the genome goes up, the number of genes goes up. And sometimes it goes up exponentially. So, for example, if you look at prokaryotes, there's this very nice correlation between the number of nucleotides they have in their DNA, the number of genes, and some of them it's kind of weird. So rice, for example, has about the same number of genes as this pufferfish, but it has about almost twice as many genes, and that's because rice actually is what we call tetraploid. So this fish is diploid, has 2 sets of chromosomes. Rice has 4 sets of chromosomes. So you see a little bit of difference there. One of the things you'll notice here is if you look at mice and humans, we have almost the same number of nucleotides, and we have almost the same number of genes. It's not a coincidence that we use mice to study things about humans, because we aren't that different. In fact, mice are probably about 95% similar to human beings. And we know that now because of the Human Genome Project. So this allows us to compare, and then when we study the genes in, say, baker's yeast, that allows us to make predictions about how those genes might work in bigger organisms or more complex organisms. So one of the things that we've looked at is if you look at the functions of human genes. Now, I'm not really going to break down this pie chart because I'm not a fan of pie charts, but one of the things you'll notice is for about 40% of the genes, we still don't know what they do. So we know the genes are there, we just don't know what they're actually doing. And the gene function is, there's a lot of variety here. There's a certain portion that are enzymes, there are cell signals, nucleic acid binding, miscellaneous. So there's a ton of different genes. And so we are just sort of scratching the surface. Now, I talked in the last video about transposons. So these are these things called jumping genes, or portions of DNA that can rearrange themselves. Transposons, by the way, some of them actually arose from viruses. So for example, we categorize the transposons based on how big they are and what kind of repeats they have. So there's this one called a long terminal repeat transposon. That actually came from retroviruses. So that came from through evolution, millions of years of evolution, infections by viruses, and that DNA got incorporated into your DNA. It doesn't get expressed because the cell basically turned off that DNA, but those DNAs are still there as transposons. Now, the other thing we discovered is that there's a lot of variation at one nucleotide within the DNA of human beings. So basically, we are filled with point mutations. Now, we call these things single nucleotide polymorphisms. Polymorphism just means different shape. Single nucleotide, it's a change in one nucleotide. And so these are very common. we see these single nucleotide polymorphisms in genes, and they actually do have effect. They're point mutations. They do have effect on the proteins that are made. And so one of the things that we found is we can use these for diagnoses. So for example, if it's a gene that allows you to metabolize a drug, these single nucleotide polymorphisms are very important, because if I have one variant of them, I can metabolize a drug very differently than someone has a different version of that SNP. We call them SNPs. So these are used in when we talk about how drugs are processed by the body. And that was one thing we didn't really know would be there when we did the Human Genome Project. So genomes, we talked about sequencing the DNA. We talked about sort of what that means with the Human Genome Project, and we talked a little bit about the results.